Hey guys, let's talk about Chapter 9, Section 3. Mitosis and cytokinesis. Eukaryotic cell division includes mitosis, which is the division of the nucleus, and cytokinesis, which is the division of the cytoplasm. During mitosis, sister chromatids are separated and distributed to daughter cells. Before mitosis can begin, the chromatin has to condense into very tightly compacted chromosomes, which can be seen using a microscope. Each species has a characteristic number of chromosomes. Humans have 46, and goldfish have 94, so you can see that the higher the number of chromosomes does not mean a larger or more complex organism. The diploid number includes 2 sets of chromosomes of each type. Humans have 23 different types of chromosomes, but we have 2 of each. That's called the diploid number. Haploid cells, which are sperm and egg, only have 1 of each chromosome. So the number of chromosomes in a sperm and egg cell are 23, whereas other body cells have 46 chromosomes. And we're going to talk about why that haploid number is important whenever we move on to meiosis, which I believe is the next chapter. During interphase, a cell prepares for cell division. We've already talked about that. The organelles are duplicated, and at the end of the S phase, we have 2 copies of the DNA. Okay, we have each chromosome has doubled, and it consists of 2 identical sister chromatids that are attached at a single point in the middle called a centromere. Okay, this is when they make the characteristic S shape whenever they are condensed, getting ready to divide. During mitosis, the centromeres holding the sister chromatids together break down, allowing those chromatids to separate and be distributed to the 2 new cells. Here we have a chromosome. There are 2 sister chromatids that are identical, linked together at the centromere. Okay, that centromere is going to break down and allow them to separate. You also see the kinetochore, which is this little orange bit right here. That is the attachment point for the spindle fibers that are actually going to move the chromosomes or the chromatids around. Just outside the nucleus, there is a structure called the centrosome. This is responsible for organizing the microtubules that help manipulate the chromatids as they move around. The centrosome was replicated during the S phase of interphase, so there are actually 2 of them. In animal cells, they contain a structure called a centriole that helps move the chromatids around. The centrosome organizes the mitotic spindle, which contains lots of fibers. Those fibers are made up of microtubules, and they form to help move the chromosomes around, and they help change the shape of the cell for cell division. The phases of mitosis. Our first phase of mitosis is prophase. This is when the chromatin condense and form that X shape that you can see through a regular microscope. Each chromosome has 2 sister chromatids that are held together at the centromere. During this prophase, the nucleolus disappears because we're not making any ribosomes. Right? We are focusing on making sure that these 2 new cells have all the right structure. So we're just getting rid of the nucleolus for a little bit. The nuclear envelope starts to break down, because remember that's what holds all the DNA in that central region. Well, if we want to move the DNA, if we want to move the chromosomes, we've got to get rid of that nuclear envelope. The spindle begins to assemble so those chromosomes can be moved around. The 2 centrosomes move away from each other. In animal cells, the microtubules form a star-like array called asters. Our next phase is prometaphase. Some textbooks don't make a distinction, they just lump this stuff in with prophase. But our textbook does. This is when preparations for sister chromatid separation is evident. The centromere of each chromosome develops 2 kinetochores, which are specialized protein complexes, and that is where the the tubules, the microtubules, will connect to be able to move those chromatids to opposite sides of the cell. Our next phase is metaphase. This is when the chromosomes are pulled around by that kinetochore and forced to align right at the equator of the cell. This is called the metaphase plate, which is an invisible line that shows the exact middle of the cell. This cell will remain in metaphase until every single chromosome is lined up exactly in the middle and the kinetochores are attached to the spindle fibers correctly. Because if they're not attached correctly, then the chromosomes won't separate correctly, and then we get cells that have too much or not enough DNA. Once that happens, once they're lined up correctly, anaphase starts. That's when the centromere dissolves and the sister chromatids are no longer attached to each other. They're now called daughter chromosomes, and they are pulled to opposite sides of the cell by that kinetochore where they're attached to those fibers. And they will move further and further apart until they are at opposite sides of the cell. Telophase, this is when those spindle fibers that did the moving, they disappear. They're not needed anymore. A new nuclear envelope forms around those daughter chromosomes. There are now two clusters of DNA of chromosomes that will become two new nuclei. They're identical. They have the exact same DNA in both of them. And a new nuclear envelope forms around that DNA. Once that new nuclear envelope has formed, then the chromatin becomes unraveled. It doesn't need to be raveled anymore. It's protected by that nuclear envelope. Here we have a diagram showing mitosis in animal cells on top and plant cells on the bottom, and an artist drawing here in the middle. In early prophase, the centrosomes have duplicated, chromatin is condensing into chromosomes, and the nuclear envelope is starting to break down. In prophase, the nucleolus has disappeared, and the duplicated chromosomes are visible. The centrosomes begin moving apart, and the spindle is in the process of forming. In prometaphase, the kinetochore of each chromatid is attached to the kinetochore spindle fiber, and polar spindle fibers stretch from each spindle pole and overlap. In metaphase, those centromeres of duplicated chromosomes are aligned at the metaphase plate, which is the equator of the cell. The kinetochore spindle fibers attached to the sister chromatids come from opposite spindle poles. In anaphase, those sister chromatids are pulled apart and become daughter chromosomes that move toward the spindle poles. In this way, each pole receives the same number and kinds of chromosomes as the parent cell. When this goes wrong, that's when we get conditions like Down syndrome, because the chromosomes didn't separate properly. Now, that would have happened in a sperm or egg cell, which would actually be happening in meiosis, but it's a similar process. In telophase, daughter cells are forming as nuclear envelopes and nucleoli reappear. The chromosomes will become indistinct chromatin, so they're starting to uncoil. Cytokinesis simply means the division of the cytoplasm, and it makes sure that there's the same amount of cytoplasm in both of the daughter cells. This is when each daughter cell is enclosed in its own plasma membrane, and it often starts during anaphase. It proceeds a little bit differently in plant cells than in animal cells. In animal cells, it's very simple. It's like pulling apart one big piece of Play-Doh into two smaller ones. Okay, something called the cleavage furrow appears between the daughter nuclei, and it's formed by a contractile ring of actin filaments. And it's pulled together and pulled together and pulled together until eventually it pinches the mother cell into two new daughter cells. In a plant cell, they can't just pinch together like an animal cell because they have the rigid cell wall. So, little vesicles made by the Golgi apparatus line up in the middle and eventually fuse into one long, thin vesicle extending the length of the cell. That forms a new cell membrane between the two. It becomes filled with middle lamella, which is like it's a substance that is found between cell walls. And the daughter cell later secretes primary cell walls on opposite sides of that little lamella, which cements the primary cell walls together. Kind of like building a brick wall through the middle of the classroom. Okay? It would separate it into two distinct rooms. Here is a diagram and an actual image of the cleavage furrow and contractile rings in an animal cell. The cleavage furrow is where it starts to pinch together. The contractile ring is what's actually causing that cleavage furrow to form. So here we have an early part of cytokinesis where they're still pretty connected. Here we have a later image where they have separated more and more and more, and you can see that they are barely holding together. Here we have cytokinesis in a plant cell. This is the cell plate. That's those vesicles fusing together to make the cell membrane, and eventually the cell wall will form. Functions of mitosis in animal and plant cells. The functions of mitosis, it permits growth and repair of the organism. In flowering plants, meristematic tissue retains the ability to divide throughout the life of the plant. Meristematic tissue are like roots and shoots. They are parts of the plant that grow incredibly quickly. In trees, this increases their girth or width with each growing season. In mammals, mitosis is necessary when a fertilized egg becomes an embryo, because we want to go from one individual cell to an organism that contains millions of cells. This is when an embryo becomes a fetus. After birth, a child becomes an adult, or when a cut heals or a broken bone mends. So new cells are added to damaged ones, or they're just increased to make the organism larger. Stem cells. Many mammalian organs contain stem cells. They retain the ability to divide indefinitely. Red bone marrow stem cells divide to produce various types of blood cells. Therapeutic cloning to produce human tissues can begin with either adult stem cells or embryonic stem cells. Embryonic stem cells can be used for reproductive cloning or the production of a new individual. Okay, so back in the 90s, scientists cloned a sheep named Dolly. Okay, that was reproductive cloning. They took embryonic stem cells from an organism and they made a new one. Here we have a diagram showing the two types, reproductive and therapeutic. So here we have reproductive. The clone is born. Okay, so we have an egg, and we basically make a copy of the organism. In therapeutic, we grow specific cells, specific type, to help repair damaged cells. And that's it. If you guys have any questions, let me know.